Okay, here next come to the third question of 2019 A-level. Okay, which of the following statements regarding microscope is correct? So microscope is also an important part. Okay, microscope means also it is also an important part in where like most of the papers it's been questioned about a one MCQ is there from microscope in most of the papers. Okay, not in recent like in 2025 I don't remember, but mostly like in most of the papers uh, about microscope there will be an MCQ. Okay, let's see. Which of the following statement regarding microscope is correct? In a light microscope, what is a light microscope? Normally in our school laboratories, we find light microscope, right? So in a light microscope, visible light is passed through the objective lens and then through the specimen. Okay, they are telling which statement is correct. So correct statement only you have to find. First you have to highlight that. Okay, what we know about a light microscope? Like if you go through your knowledge, what do you know about a light microscope? So how in that visible light is passed? We know that visible light is passed to the specimen and through the glass lens, right? So visible light is passed to the specimen and through the glass lens. That's how the light microscope is there. Okay, so that way the first one is wrong. Projections of a light beam through a vacuum is the principle of an electron microscope. So what is the principle of an electron microscope? Now here, what is the simplest microscope? It's a single lens, no? So what do you know about electron microscopes? Let's see. Electron microscope mean, what is it? There are two types of electron microscope. That is transmission electron microscope and then scanning electron microscope, which you call as SEM and TEM. So these are two types of electron microscopes. And what is electron microscopes? So their resolution power is inversely proportional to the wavelength. You know that? No? So how do you, in the normal microscope, it is like, it's a different story, but in electron microscopy, a beam of electrons only focus to the specimen to its surface, no? So, and then like electron microscope, it helps us to magnify the object 1 into 10 to the power 8 times, no? That's in theory. And in practice, how much does it magnify? 5 into 10 to the power 5 times. These are the things that we know about electron microscopes. Okay. So, and then what are they telling is, projections of light through a vacuum is a principle of light beam. No, that's definitely wrong. No, we are not projecting light beams. We are using electron. No, electron is only being, the beam of electrons are only being focused. So second one is wrong. Scanning electron microscope is used to study the internal structure of cells. So scanning, and now we know that transmission electron microscope is there and scanning electron microscope is there. These are two types. So what do we use this TEM? I mean transmission electron microscope for? That is for, we use that to identify, see the structure, to study the internal structures of the cells, no? So that way, scanning electron microscope, not that, that is wrong. Transmission electron microscope is only used to study the internal structure of the cells. So that also we know. That's that way. The third statement is also wrong. So that should be TEM. TEM is only used to study the internal structure of the scale of a cell. So why do we use the scanning electron microscope, dear children? We use scanning electron microscope for what? Is to observe the surface view in 3D. I mean, in 3D appearance, like in three-dimensional way, we are going to, observe the surface. Only we use this microscope. Okay. So transmission, the fourth one is transmission electron microscope is used for detailed studies of living specimens. No? Okay, children. So magnification and the resolution power important. Yes, definitely. properties of all microscopes. Yeah, fourth question is, we are not using transmission electron microscope is not used for detailed studies of living specimens, no? Here electron microscope, yeah, not using for the living ones. Uh, the fifth one is correct. Magnification and the resolution power are very important properties of all microscope that we know, no? Okay.